Okay, before discussing the process of mitosis and meiosis, let's first define and discuss what a chromosome molecule is. But before we go deeper and see what a chromosome is and its function, let's recall some basic information about our DNA. So DNA, which stands for deoxyribonucleic acid, is a type of biological molecule which is used to store genetic information in the body. Now, our DNA contains double helix structure, and this means two anti-parallel single-stranded DNA molecules are actually joined together to form our helical structure, which is known as our double helix. Now, if we take a single DNA and extend it over a linear fashion in a straight line, our DNA molecule will have a length of approximately 1.5 meters in length, which is equivalently to over 5 feet in length. Now the question is, how does the cell manage to do that? How does the cell manage to take such a long biological molecule and place it in a very small region of the cell known as nucleus? In fact, our body is capable of storing 46 of such a long biological molecule and place it in a very small structure or a very small region of the cell known as our nucleus. How does the cell actually manage or accomplish to do this task? Now, what the cell actually does is it takes such a long biological molecules and it wraps it around a special protein known as histone. So it uses a special protein known as histone and it wraps around our DNA molecule, our double helix structure for DNA molecules, and it puts it or condenses it in a very, very condensed structure known as our chromosome. Now, chromosome is predominantly composed of our protein and the rest is DNA, but a very, very small portion of our DNA molecule is also made up of our RNA. So let's actually see how does actually cell manage to take such a long biological molecule and they put it in a very condensed structure known as chromosomes. What are the process actually our cells takes to accomplish this task? So let's go and let's have a look on this. Okay, so this is our structure of our DNA, which actually describe how our cell is managed to take such a long biological molecules and condense it into a very small region known as our chromosomes. So this is the double helix structure for our DNA. As we zoom out, the structure becomes smaller and smaller. So if we start from here, as we go down, the structure of DNA actually becomes smaller and smaller as we zoom out, and it reaches until we stop at this region here, where actually our DNA looks like a single line. And at this region, what actually cells does is it uses special type of protein known as histone, and it wraps around our histone this double helix structure. So we have this special protein, and around this special type of protein known as histone, our double helix structure for DNA is actually wrapped around. So this is actually what happens at this region here. Now, next to this is our structure known as another structure known as nucleosome, and the nucleosome is nothing much but 8 of this histone. Now, what our cell does is it takes 8 of this histone and put them together and pack them together on top of one another and form a single structure known as nucleosome. So nucleosome is nothing much but 8 of this histone which are now grouped together to form another structure known as nucleosome. So as we said, as we move out, our structure becomes smaller and smaller and until we reaches at this region here. And what actually happens at this region here is our nucleosomes are now packed together on top of one another in a helical fashion, in a helical structure known as solenoid. So solenoid is nothing much but this nucleosome, which are now packed together in a helical structure to form another structure known as our solenoid. Now this doesn't stop here. Our solenoid coil even further to form another coils known as our supercoils. So solenoid coil even further to form another structure known as our supercoil. Now this goes until we reaches another region here known as chromatin. Now at supercoils, what actually cells does after formation of our supercoils, it is our cells actually what it does is it packs this supercoil on top of one another. It packs together these supercoils on top of one another to form a single structure known as chromatin, which looks something like a straight line as we can see here. So this is a chromatin, which is now our supercoil, which are now packed on top of one another to form another condensative structure known as our chromatin molecule. Now chromatin, the same as our chromosomes, is mainly composed of protein. It's made up of protein, and the rest is DNA. And also a very small portion of our chromatin is made up of our RNA. Now it doesn't end here. Actually, our chromatins, they can coil even further and further to form a final condensed structure of our DNA molecule, which is now known as our chromosome. So chromosomes, now we come to conclude that these chromosomes is nothing much but a very, very condensed structure of these DNA molecules, which is wrapped around our special protein known as histone. And the chromosomes is predominantly composed of protein. The rest is DNA, and a very small portion of our chromosome molecule is actually made up of our RNA. And these two chromosomes are actually joined together by special a protein in the region known as centromere. So in this region here known as centromere, special type of protein are actually used to join together our two chromosomes. Now another thing which I also wanted to explain here is this concept of homologous pair. Now if two chromosomes which are joined together contain genes which code for the same trait or similar trait, such as hair color or eye color, height, and etc., these two chromosomes are known as homologous pair. So homologous pair, this is a pair of chromosomes which contain genes which code for the same trait or same character, such as hair color, eye color, height, and etc. Now, when our chromosomes are in homologous pair like this, which are now two chromosomes joined together, this state is also sometimes known as our diploid state. So diploid state is when our two chromosomes are joined together, but when our chromosomes are not in pair, like you see in diagram here, this is actually known as an haploid state. And some cells actually exist mainly in haploid state. And the example of those cells which exist in our haploid state are germ cell or gamete. So this is short, briefly introduction of our chromosomes. As we said, the chromosomes is nothing much but a very, very condensed structure of our DNA molecules. Because our DNA molecules, these are biological molecules actually, which are very, very long in length. So it is actually very difficult to store such a long biological molecule in a small region like our nucleus. So they say that what it actually does, it takes such a long biological molecules and it wrap around a special type of protein known as histone, and it put it together on top of one another. It coalesces it into a very, very, very condensed structure known as our chromosomes. And our chromosomes, as we said, is predominantly composed of protein. The rest is DNA, and a very small portion of our chromosomes is actually made up of our RNA. So next video, we're going to see much more details about our cell cycle and the process and how this process of mitosis and meiosis actually take place. Okay, thank you.